Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive into today's topic, please take a moment to like this video and subscribe to our channel. It really helps support the Science Experience and allows us to bring you more fascinating discussions. Okay, so today we're plunging into a truly counterintuitive puzzle within the science of aging. We're talking about mosaicism in our tissues, specifically how tiny scattered patches of dysfunctional cells might actually be more damaging than if an entire organ were just uniformly a little less efficient. Think about that for a second. Why would a few bad apples spoil the whole barrel more effectively than if every apple were just slightly bruised? That's our mission today, to unpack why this patchwork problem might be worse for your health than a more widespread but milder decline. Okay, let's unpack this intriguing question.
Yeah, it's a concept that really flips our intuition on its head, doesn't it? When we talk about this mosaicism, we're drawing from some really compelling evidence about mitochondrial oxidative phosphorylation deficiency, or OXPHOS deficiency. Now, if that sounds like a mouthful, let's simplify. OXPHOS is basically the powerhouse process. It's how your mitochondria, those tiny crucial engines inside, well, almost every one of your cells produce ATP. That's the vital energy currency your body needs to function. And it's absolutely critical for organs with really high energy demands, like your heart, your brain, your muscles. So in this context, mosaicism isn't about a gradual, even slowdown across all your cells, not at all. Instead, it describes the development of these discrete, isolated patches of cells that become severely deficient in OXPHOS, while their immediate neighbors might remain relatively normal. What's truly fascinating here, and kind of worrying, is how this uneven distribution can have such a disproportionate and often devastating impact on overall tissue function.
That immediately sparks a question in my mind. Where do these problematic patches even come from? I mean, are cells just randomly going rogue, or is there a specific process at play here?
It really starts deep inside the cell with our mitochondrial DNA, or mtDNA. So unlike the DNA in your cell's nucleus, mitochondrial genomes are particularly susceptible to mutations. They have a limited repair capacity, and they're constantly exposed to reactive oxygen species, ROS, which are essentially metabolic byproducts from the very energy production process itself, sort of an occupational hazard for mitochondria. So as we age, these somatic mutations, especially things like large-scale deletions in the mtDNA, they accumulate in a pretty random manner within individual cells.
Okay, random mutations, but how does that lead to distinct patches?
Right, so the story doesn't end with just random damage. There's a crucial next step called clonal expansion. Imagine a single cell. It starts accumulating more and more of these mutant mtDNAs. Now, for a long time, maybe nothing much happens visibly, but eventually it hits what scientists call a phenotypic threshold. Think of it like a dimmer switch for that specific cell's energy production. For a while, even with some bad wiring, the mutant mtDNA, the light, the cell's function, seems okay. But once that bad wiring crosses a certain point, say above 60% of all the mtDNA in that particular cell, then suddenly that dimmer switch goes from maybe a subtle dimming to flicking the light right off. That's when overt respiratory defects, the OXPHOS deficiency, actually manifest. And that leads to the formation of distinct localized patches of dysfunctional cells. We've seen this really clearly in advanced mouse models like the K320E twinkle mouse heart model, where only a small fraction of heart cells actually become COX negative, severely OXTOS deficient. And the amazing and frequently unsettling thing is that the cells right next door can be completely spared, just functioning normally.
Okay, so it's not just random damage spread thin, but more like a single bad seed taking root and then multiplying its dysfunction within a very specific area, almost like islands of failure. It's like having a few critical circuits fail spectacularly in a finely tuned machine, rather than just a general power sag across the board. That really makes you think. Now, this is where it gets really interesting for me and seriously counterintuitive. Why on earth would these scattered patches cause more trouble than if the entire tissue just had a uniform mild decline? My gut still tells me a whole organ being a little less efficient would be way worse than just a few bad spots.
That's the absolute crux of it. You're right. If an entire tissue, let's say a muscle, has a uniform, mild mitochondrial impairment, think of it like a sports team where everyone is just a little bit tired, maybe they all slow down slightly. They can often still coordinate adaptive compensatory mechanisms. They might all adjust, perhaps globally reducing their workload, but they still function as a cohesive unit, more or less. The system is weakened, sure, but it's still somewhat synchronized.
Okay, that makes sense. A coordinated slowdown.
Exactly. But in stark contrast, mosaicism creates these focal areas of severe energy failure right next to perfectly healthy cells. And this prevents any kind of harmonized, tissue-wide adaptive response. This cellular heterogeneity, this patchiness, profoundly disrupts intercellular communication, the vital chatter between cells. It messes up the concerted regulation of ionic balances like crucial calcium handling and even paracrine signaling, which is how cells talk to their neighbors through secreted molecules. Take the heart, for instance. Patchy loss of OXPHOS leads to these localized disturbances in calcium handling, which significantly predisposes the heart to electrical conduction abnormalities and potentially very dangerous arrhythmias. It's like having random dead spots pop up in a highly synchronized electrical grid. It causes chaos.
Wow, that's fascinating. So these bad patches aren't just dead weight. They're actively messing with their neighbor's ability to function properly. And thinking about the heart, its electrical system is incredibly sensitive, right? Even tiny disruptions can have huge consequences. So what are these hot spots actually doing to actively cause problems?
You've hit on a critical point there. Cells with severely impaired OXPHOS don't just produce less ATP, less energy. They also often generate excess reactive oxygen species, ROS. These are essentially those damaging metabolic waste products we mentioned. This leads to localized oxidative damage and activates inflammatory pathways specifically in these focal areas.
Ah, so they become sources of stress themselves.
Precisely. These hotspots of oxidative stress can severely exacerbate tissue damage. In the heart, for example, ROS-induced modifications can further compound the risk of arrhythmias, but it goes even deeper than that. This localized damage can trigger something called a senescence-associated secretory phenotype, or SASP. These senescent cells, the bad apples, they don't just sit there quietly failing. They start spewing out a cocktail of inflammatory and damaging factors that actually propagate dysfunction to adjacent, previously healthy cells. It creates this vicious cycle where a small patch of dysfunctional cells actively poisons and corrupts its neighbors.
Whoa. So they're not just dysfunctional, they become active saboteurs, almost recruiting healthy cells into dysfunction. That fundamentally changes how we might think about cellular health and aging, doesn't it? Especially when you consider how crucial electrical signals are in the body.
Absolutely. The body's electrical systems, particularly in the heart, are just exquisitely sensitive. Research shows that even if only a tiny fraction of heart cells become OXPHOS deficient, we're talking maybe as low as 0.6% in one mouse model, this mosaic pattern creates regional disparities in how electrical signals propagate. This really sets the stage for severe arrhythmogenic events like premature ventricular contractions or blocks in the electrical pathways, the AV blocks.
So a tiny fraction causes major problems. How?
Well, the critical difference is this. A uniformly affected tissue might globally adjust to slower conduction. It's not ideal, but it could potentially preserve overall electrical synchrony. Imagine an orchestra playing a piece slowly, but everyone is still in perfect time together.
Right. Slower but synchronized.
But the juxtaposition of healthy and compromised cells in mosaicism completely disrupts this delicate balance. It's like having a few musicians in the orchestra suddenly playing wildly out of sync or maybe dropping out entirely while the rest desperately try to carry on. It leads to life-threatening rhythm disturbances precisely because of these unpredictable electrical discontinuities. The sharp contrast is the killer.
Okay, that makes the danger much clearer. What about repair? Does this patchiness affect healing too?
It certainly does. Think about tissue regeneration and repair. Your body is constantly fixing itself, right? But this process relies heavily on a healthy microenvironment and really coordinated cell-to-cell interactions. Mosaicism introduces this functional heterogeneity, this patchiness, that directly impairs these vital regenerative signals. In skeletal muscle, for instance, patches of OXPHOS deficiency disrupt the crucial interactions between satellite cells, those are your muscle stem cells, and their surrounding support niche. This leads to asynchronous regeneration, meaning the repair process happens unevenly, inefficiently, and results in abnormal muscle fiber maturation.
So the repair crews can't work properly because the foundation is uneven.
That's a great way to put it. This irregularity not only compromises the body's natural repair mechanisms, but also predisposes the tissue to things like fibrosis, which is scarring, and fatty degeneration. Both further severely impair function. It's like trying to rebuild a house where some of the new bricks are faulty and actively undermining the structure around them. The whole repair process gets derailed.
That's a powerful picture, not just damage, but active sabotage and hindered repair. So how does this patchwork problem manifest in some of the body's most vital organs? It sounds like the heart, brain, and muscles are particularly vulnerable given their high energy demands.
They absolutely are. In cardiac tissue, as we touched on, studies have shown that even a small minority of cells, less than 1% sometimes, with high mtDNA deletion loads, existing as isolated patches, are strongly linked to significant cardiac disturbances. Again, this isn't just about a generally weaker heart. It's about these erratic, potentially life-threatening rhythm problems like spontaneous ventricular premature contractions and AV blocks. And unlike a uniform decline where the heart might try to adapt evenly, maybe by boosting mitochondrial numbers everywhere, in mosaicism, these compensatory responses are spatially limited. They simply can't prevent those localized conduction disruptions that directly drive the arrhythmias.
Okay, so the heart's electrical system is uniquely vulnerable. What about the brain?
Well, the central nervous system, our brain, is incredibly energy demanding, constantly working. Focal OXPHOS deficiency in neural tissue can severely compromise neurotransmitter balance, disrupt synaptic connectivity. That's how brain cells talk to each other, and impair calcium buffering, which is crucial for preventing cell damage from overexcitation. This localized energy failure can actually initiate damaging cascades similar to those seen in devastating neurodegenerative disorders like Alzheimer's and Parkinson's. In those conditions, regional mitochondrial dysfunction is thought to exacerbate the buildup of harmful proteins and promote neuronal death. And because the damage is patchy, a uniform neuroprotective response from the brain is hindered. It's much harder to target repair efforts effectively when the problem isn't spread evenly.
And muscles. You mentioned stem cells and repair.
Yes, skeletal muscle also faces unique challenges. Muscle tissue naturally has different fiber types, fast twitch, slow twitch, which is its own kind of healthy mosaic. But when these pathological OXPHOS deficient patches accumulate, this natural healthy organization gets disrupted. It compromises not only the contractility of individual muscle fibers within the patch, but also the coordinated function of the entire muscle. As we discussed, this patchiness interferes with regeneration after injury and ultimately leads to progressive muscle weakness and atrophy over time. Imagine a team of rowers where a few oars are just broken or completely out of sync. The whole boat suffers. Its speed and coordination are shot, even if most of the rowers are strong.
That makes sense. Are other organs affected too?
Oh yes, this isn't just limited to those three major players. Similar principles seem to apply to other organs too. We see evidence suggesting links in the liver, potentially leading to issues like steatosis or fatty liver disease and fibrosis or scarring. And in the kidneys, it could disrupt their vital filtration capabilities by creating localized failures. In pretty much each case, the patchy distribution seems to mean that any compensatory mechanisms the body tries to mount simply fail to sustain normal physiology. This likely accelerates both aging and disease susceptibility far more quickly than if the decline were uniform and maybe more manageable for the system as a whole.
This really paints a picture of localized chaos being worse than generalized slowdown. So what does this all mean for us, the listener? What's the big picture takeaway? And maybe how should we think about aging differently now?
Yeah, I think the salient point emerging from all this literature is pretty profound. It's that mosaic OXPHOS deficiency creates these localized hotspots of severe dysfunction that the surrounding tissue is inherently unprepared to compensate for. It's not equipped to handle these sharp gradients in function. Unlike a gentle uniform fading across an entire system, which might allow for gradual adaptation, this patchiness initiates these really maladaptive cellular stress responses. Things like that localized ROS accumulation, the inflammation, and that vicious SSP cellular senescence we talked about, they actively propagate dysfunction to nearby healthy cells. It's like a tiny, uncontrolled fire that rapidly spreads and damages the neighborhood.
Spreading the damage actively.
Right. And it profoundly disrupts the intercellular communication networks that are absolutely vital for synchronized tissue function. Think again about the precise electrical signals in the heart or the delicate neurotransmission patterns in the brain. Mosaicism creates these islands of dysfunction that compromise the integrity of the entire network. The whole system becomes less reliable. Furthermore, as we saw with muscle, this mosaicism severely compromises the tissue's capacity for repair and regeneration. These dysfunctional islands create these microdomains where the necessary signals for stem cell activation and proper differentiation are either insufficient or maybe even actively harmful. The repair process just breaks down locally.
So summing it up,
The key takeaway really is that the patchy nature, the heterogeneity of mitochondrial dysfunction seems to overwhelm the body's natural capacity for compensatory adaptation. And this leads to a cascade of pathological events, the ROS, the inflammation, the SASP, the communication breakdown, the failed repair, that culminates in disproportionately severe organ dysfunction and accelerated decline. It appears far worse than if an entire organ were just uniformly moderately impaired. It's the unevenness, the dyssynchrony, that causes the most profound damage.
That's a truly powerful thought to end on. It really suggests a potential shift in how we might approach age-related health problems, which raises an important question, maybe for future research. What if focusing on restoring uniform mitochondrial function, maybe making sure all cells are declining at a similar, manageable rate, rather than just boosting total function overall, becomes a key target for future aging interventions? Something to think about. Thank you for joining us for this deep dive into cellular mosaicism and aging. It's certainly been an eye-opening discussion for me. If you found this conversation as intriguing as we did, please do remember to like this video and subscribe to the channel if you haven't already. It really helps support the SANS Science experience and helps us continue exploring the wonders and sometimes the counterintuitive puzzles of science with you. Until next time, keep asking those big questions.